Hello friends, this video on applications of biotechnology part 9 is brought to you by examfear.com. No more fear from exam. Now the question is, these cells, these lymphocytes are also mortal, that is they are not going to live forever. Therefore periodic infusion of these genetically engineered lymphocytes are needed. But if this is done at an early embryonic stages, then it could become a permanent cure, because over a period of time, now a couple of times if this infusion is being done, then what happens is your lymphocytes will give rise to more cells, like how it happened in case of genetically engineered insulin. Now, so these cells, when they multiply, they will also produce more cells, and they all will have the desired gene for ADA production. So that way, the gene therapy can become a permanent cure to ADA deficiency. However, this could be a cure only when it is detected at an early embryonic stage, because later when a person already is, like say he is an adult, he or she is an adult, and that time this kit could not be a permanent cure, because that time your body is all developed. So it will take time. In fact, it might not be that successful. It might not be a permanent cure at that time. But at a very initial stage, yes, gene therapy can actually replace the missing gene which is responsible for the production of ADA enzyme. So now the third important application, that is molecular diagnosis. Now, diagnosis of any disease is very important, because until and unless the disease has been diagnosed, treatment cannot be given. Now, there are many deadly diseases, for example, cancer or AIDS, where early detection is extremely significant, because if there are certain types of cancer which when treated at, which when diagnosed at a very early stage can be completely treated. But if it goes to a later stage when it has spread throughout the body, then the treatment becomes difficult. So early detection of a disease is extremely useful for treatment of the disease. So this concept of genetic engineering also helps in early detection of disease, and thus ensures effective treatment of the disease. Now what happens is, how do we diagnose a disease? Now any disease is generally caused by some pathogen. So if we talk about the infectious diseases, they are caused by the pathogen. Now mostly how do we diagnose disease? With the help of blood test or urine test. Now when a pathogen is present in a very high concentration, when a large number of pathogens are already present in the body, only then the symptoms of that disease are seen. And when the symptoms are seen, only then what happens, your blood test or urine test also comes up with their result. So these kind of tests, which we normally do, they all can detect a disease only when the concentration of the pathogen is quite high inside the body. So these kind of tests cannot be used for early detection. However, in some diseases like AIDS or cancer, early detection is very much significant. So therefore, there are few techniques which can help in detecting a disease even when the concentration of the pathogen is extremely low. Now, some of these tests are PCR test, where PCR stands for polymerase chain reaction. We discussed polymerase chain reaction in our previous lesson on biotechnology. The other test is the ELISA test, where ELISA stands for enzyme-linked immunosorbent assay. This ELISA test is used for diagnosing AIDS at a very early stage. So now we will look at these two tests and how do they help in early diagnosis. So let us first discuss the PCR test. So here in this test, very low concentration of pathogens can be detected by amplification of their nucleic acids. Now if you remember, or if you can recall the PCR test, where we talk about the polymerase chain reaction, what did we see? We saw that a small concentration of anything can be amplified. When we say amplification, what do we mean? That means multiple copies of that particular DNA can be created. So you can take, for example, this PCR was done as a part of amplification of the gene of interest. So while following the process of genetic engineering, we try to amplify the gene of interest in terms of making multiple copies of the gene of interest. So that is known as amplification in terms of PCR. So here also, even if the pathogen is present in very small concentration, using this technique, that small concentration can be amplified because their nucleic acids or their genetic material can be amplified by creating multiple copies. So that's what is PCR, where a chain reaction takes place. So from one single DNA, you can create 2, from 2 to 4, from 4 to 8, from 8 to and so on. So that's how the chain reaction keeps on continuing. So finally, you actually get an amplified version of the pathogen. And that's how PCR is able to detect the presence of that pathogen, even though the pathogen is present in extremely low concentration. So this is how the PCR reaction take place. So you start with one single DNA and you end up having 2. And again from 2 you end up having 4. And again from each of them you will have 2. Again from each of these you will have 2 copies. So over a period of time, you actually end up having so many copies, and that is known as the amplification of the genetic material, or amplification of the nucleic acids. Now, this reaction or this test is done regularly on AIDS patients to detect the presence of HIV, because even if that HIV, which is nothing but a virus, even if it is present in extremely small concentration, this test can actually amplify, or it can create multiple copies of that virus, and it can give you a signal stating that yes, this virus is present. Similarly, it can be done on patients who might suffer from cancer, or who think that since they have some genetic record of cancer, so they might suffer from it. So to detect mutated genes, if any, present inside their body. It can also be used to detect other sort of genetic disorders, where you just want to check if any of the gene is mutated, or if there is presence of pathogen in any of the part of your body. So that way PCR test is extremely useful, and this concept of PCR came from genetic engineering. Thank you. Please visit www.examfeo.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.